Assalamualaikum warahmatullahi wabarakatuh, and hello everyone. We are Group 3 and today we will present our Benthos sampling report. My name is Nur SAFIYA binti MOHAMMAD HAZUAN, metric number A211552. Benthos sampling serves as a key biological monitoring technique for evaluating the ecological condition and water quality of rivers and streams. Benthic macroinvertebrates function as effective bioindicators because they live at the bottom of aquatic habitats and respond differently to environmental changes and pollution levels. In this fieldwork, Benthos sampling was performed at Pangsun Hulu Langat, Selangor, at two stations in the same stream. Station 1 was located in an open area, while Station 2 was situated in a shaded area. The collected organisms were identified to the family level and analyzed using biological indices, including the EPT index, ASPT index, BMWP, and Shannon-Winner index. We also measure water quality parameters such as pH, dissolved oxygen, temperature, and conductivity. These measurements help us compare the ecological health and water quality between the two stations. There are three objectives in this study. The first objective is to determine the water quality of Station 1 and Station 2 based on pH, dissolved oxygen, temperature, and also conductivity. The second objective is to identify and compare the benthic macroinvertebrate families found at both sampling stations. The third objective is to assess the ecological condition of both stations using the EPT index, ASPT index, BMWP, and Shannon-Winner index.
Thank you, ALEEYA, for the introduction. Hello, I am SOLEHAH bin JAMALUDDin A209527, and we will now be moving into our materials and methods. To collect and process our samples, we utilize standard field equipment as shown in the slide, including a stirrer net, sieves, trays, and sorting tools, as well as 70% ethanol to preserve our specimens. For our in-situ environmental parameters, we use a digital handheld probes to measure dissolved oxygen, pH, and temperature, while a GPS unit was used to map our exact coordinate. Our fieldwork was conducted upstream at Pangsun in Hulu Langat, Selangor. The site sits at an elevation of 112 meters above sea level. Geographically, the upstream section is narrow and turbulent, heavily dominated by large boulders and angular rocks, which provide an ideal habitat for benthic macroinvertebrates. We sample across two distinct stations, an open area and a shaded area. To collect the benthos, we use the kick method to disturb the riverbed substrate, allowing the current to flush the organisms into our stirrer net. We also carefully rinse large rocks over a sieve to catch any clinging species, transferring everything into Ziploc bags using pipettes. Samples were then spread onto trays, and individual organisms were isolated into petri dishes. Using a magnifying glass and taxonomic field guides, we identified the organisms down to the family level, recorded their abundance, and preserved them in ethanol. Finally, for our data analysis, we first calculated the water quality index using our three parameters. Next, we computed four biological indices, Shannon, EPT, BMWP, and ASPT, from our family count data. By combining these quantitative index scores with our qualitative field observations, we were able to comprehensively evaluate the overall ecosystem health of the river. Thank you. I will now pass the floor to the next presenter to discuss our results and findings.
Thank you, Soleha, for presenting about the materials and methods. My name is LOSHINY and my matrix number is A208966. Now we'll move to the results and discussion. The water quality parameters measured at both stations showed only slight variation, indicating that Sungai Pangsun has relatively stable water conditions. For pH, Station 1 recorded a slightly higher average pH, which is 7.60, than Station 2, which is 7.42. Both values are close to neutral pH, indicating that the river water is neither strongly acidic nor alkaline, which is suitable for most aquatic organisms. Next is for the dissolved oxygen. Station 1 had a slightly higher average of DO, which is 6.30 ppm, compared to Station 2, which is 6.01 ppm. Both values are above 5 ppm, suggesting that the river is well oxygenated and capable of supporting a diverse community of aquatic macroinvertebrates and fish. For temperature, the average water temperatures were very similar, with 25.27 degrees Celsius at Station 1 and 25.23 degrees Celsius at Station 2. These stable temperatures are typical of shaded tropical streams and help maintain adequate dissolved oxygen levels. Overall, both stations exhibit good water quality with near neutral pH, sufficient dissolved oxygen, and stable temperature. These conditions are favorable for aquatic life and are consistent with the relatively high EPT and ESPT values obtained during the biomonitoring assessment. Although Station 2 had a slightly lower dissolved oxygen concentration, The difference was minimal and did not appear to negatively affect the diversity of pollution-sensitive macroinvertebrates. The EPT index is a biological indicator used to assess river water quality based on the presence of three pollution-sensitive insect orders, which is Ephemeroptera, Plecoptera, and Trichoptera. A high EPT value indicates better ecological health. At Station 1, only Hydrophyscidae representing Trichoptera and Pelidae representing Plecoptera were found. At Station 2, Hydrophyscidae, Petidae, Pelidae, and Potamantidae were recorded, indicating a greater diversity of EPT taxa. This suggests that Station 2 has a better ecological condition and supports more pollution-sensitive macroinvertebrates than Station 1. This slide shows the EPT index and the percentage of EPT taxa for both sampling stations. Station 1 recorded an EPT index of 2, while Station 2 recorded an EPT index of 4, indicating a higher diversity of pollution-sensitive taxa at Station 2. The percentage of EPT taxa was 61% at Station 1 and 63% at Station 2. This result suggests that Station 2 has a slightly better ecological conditions and water quality, as it supports a greater proportion of sensitive macroinvertebrates compared to Station 1. This slide presents the ASPT or average score per taxon for both sampling stations. ASPT is calculated by dividing the total BMWP score by the number of scoring families. Station 1 recorded an ASPT value of 5.4, while Station 2 recorded a slightly higher value of 5.7. Higher ASPT values indicate better water quality because they reflect the presence of more pollution-sensitive macroinvertebrates. Therefore, the result suggests that both stations have good ecological conditions, with Station 2 showing slightly better biological water quality than Station 1. For discussion, based on the EPT index, Station 2 recorded a higher value compared to Station 1, indicating a greater diversity of pollution-sensitive taxa such as Ephemeroptera, Plecoptera, and Trichoptera. This suggests that Station 2 has slightly better ecological conditions and water quality. For the percentage of EPT individuals, Station 2 recorded 63% compared to 61% at Station 1. This shows that both stations are dominated by pollution-sensitive organisms, although Station 2 has a marginally higher proportion. Finally, for ASPT, Station 1 recorded a value of 5.4, while Station 2 recorded 5.7. Both values fall within the range of 5.0 to 6.0, which indicates good biological water quality with a relatively low organic pollution. Overall, both stations show good ecological condition, with Station 2 being slightly better.
Now, I will present the result of Shannon-Wiener Index and BMWP scoring system. First, the Shannon-Wiener Index is a diversity index used to measure the biodiversity of a benthic community. It takes into account two key concepts: species richness, which is the number of different taxa, and species evenness, which is the distribution of individuals among taxa. The formula is shown below. Now, let's look at the detailed calculation for both stations. For Station 1, the total individual collect was 18 with 5 taxa identified. The calculation gave us H prime equal 1.38. For Station 2, the total individual collect was 33 with 7 taxa identified. The calculation gave us H prime equal 1.85. Based on the interpretation skill, both stations fall into the moderate stress category. Next, I will present the BMWP Biological Monitoring Working Practice Score. This is a biological scoring system that assigns a sensitivity score to each microinvertebrate family based on its tolerance to pollution. The BMWP score for each family was determined using the scoring system provided in the appendix. For Station 1, the total BMWP score was 27. For Station 2, the total BMWP score was 40. The interpretation skills show that both stations fall in the polluted category. In summary, for the Shannon-Wiener Index, Station 2 recorded a higher H prime value of 1.85 compared to Station 1 with 1.38, indicating greater diversity and better ecological condition. Both stations fall into moderate stress category, suggesting moderate environmental disturbance. For the BMWP score, Station 2 recorded a higher score of 40 compared to Station 1 with 27, indicating better water quality. Both stations fall into the polluted category. This is mainly because we found only a few families. However, it's important to note that families we define are actually sensitive to pollution, which is a good sign. In conclusion, the Benthos sampling fieldwork demonstrated that both stations exhibited relatively healthy ecological conditions. However, Station 2 displayed slightly superior water quality compared to Station 1. Station 2 achieved higher biological index values, including the EPT index, percentage of EPT taxa, ASPT, and Shannon-Winner index. This reflects greater macroinvertebrate diversity and a higher proportion of pollution-sensitive taxa. The water quality parameters at both stations were suitable, with near neutral pH, moderate dissolved oxygen, and stable temperature. These findings indicate that Station 2 has better habitat quality and lower environmental stress. Overall, benthic macroinvertebrates were effective indicators for evaluating freshwater ecosystem health.
This is the appendix that we use for the BNWP scoring system.
This is the references used in our study. That is all from us. Thank you.